Today we will see the gene regulation in eukaryote. So in eukaryote, gene regulation actually happens in three levels. They are transcriptional level control, processing level control, and translational level control. So in the first level, that is transcriptional level control, it is a mechanism which determines whether a particular gene can be transcribed or not. So whether a gene will be transcribed into an mRNA or not, that is which is determined in transcriptional level control. And the next processing level control, in which once the mRNA is formed, that is called the pre-mRNA, and that mRNA, the pre-mRNA, the premature mRNA is being processed to a functional mRNA. So that is the processing level control. That is all about the processing level control. And next is the translation level control. It is the mechanism which determines whether a particular mRNA is actually translated, and if so, how often and how long it is being translated. So these are three levels of gene regulation in eukaryote. So we will see the first one, transcriptional level control. So in transcriptional level control, as we have already seen the transcription factors, there are transcription factors are involved which helps in the process of transcription. So mainly there are two types of transcriptional factors are there. They are general transcription factors which bind to the promoter sequences or the core promoters in a gene, and which are which are named as TF2A, TF2B, TF2D, TF2E, F, and H. And the specific transcription factors are there, which are the factors which actually bind to other regulatory region other than promoters, known as enhancers. These transcription factors can either initiate or either stimulate or inhibit a transcription. So if it stimulated transcription, it is called activators, and in the opposite case we call it repressors. So here we can see how a transcriptional level control is being attained, or by showing a picture of a gene which is the phosphoenol pyruvate kinase or PepsiK gene, and this is the structure of gene. And here we can see this this is the promoter sequence. We can see the Tata box here. CAT and GC boxes are also there. This is the promoter sequence of PepsiK gene. PepsiK gene is or which produces an enzyme known as phosphoenol pyruvate carboxy kinase, and which is actually an enzyme involved in pyruvate synthesis. Okay. So here we can see this the Tata box, GC, and CAT boxes are there, and how these removing these regions, the Tata, the GC, CAT box, and other sequences in between these Tata and GC boxes can actually affect transcription rate. So here we can see that the first case where the gene is intact, so that the transcription rate is 100%. So that is, it is been shown that the relative promoter activity, relative promoter activity is 100% when the gene is intact. And when we remove, if we cut this Tata box from the gene, what happens is that the efficiency of transcription has been reduced to 38%. And if we remove any intervening portions between the GC and data box, not much change in transcription happens. Now it is still 95%. And if we remove GC box, this is the region of GC box. If we remove GC box and all other portions are intact in a gene, the transcription has been found to reduce to 14%. So that a major effect in the efficiency of transcription has been shown when we remove these GC boxes. So here we can interpret from this is that even the data box, removing data box, it is been reduced to 38%, and removing GC box, the transcription has been reduced to 14%. So it is more important than the data boxes. So if we remove the CAD box, what happens is 50% of transcription is being affected. And if we remove any other portions which is not in between this, which is away or which is beyond this promoter sequence, nothing happens to the percentage is in 114%. So actually the efficiency being increased in this case. So this actually shows how this mechanism or these removing these major portions in a promoter sequence can affect transcription. So next we will see the same enzyme, how this phosphoenol, as we have already seen, this already mentioned phosphoenol pyruvate carboxykinase or PepsiK, which is one of the key enzyme in gluconeogenesis, which converts pyruvate to glucose. So that is the enzyme synthesized in liver. Actually, how it affects hormone synthesis, or sorry, how it affects the protein synthesis. So we have seen that many hormones actually affect the protein synthesis, like this insulin, then growth hormone. There are several factors, or cortisol. These are many hormones which actually controls or have an effect on this, has an effect on this protein synthesis. So here we can see that this PepsiK gene, that is expression of this PepsiK gene, are affected by insulin, thyroid hormones, glucagon, and glucocorticoids. So these PepsiK gene, here we can see it is a picture which showing this. This is the PepsiK gene. Okay, so how these the other hormones like cortisol, or we call it corticosteroid, glucocorticoids, actually stimulate this PepsiK gene for protein synthesis. So we will see that. So here we can see this is the cortisol, which is seen in the extracellular fluid, and this is the plasma membrane. So this cortisol, when it enters the cell through the plasma membrane, there is a glucocorticoid receptor is here. This are the blue color which indicates the protein. So this is a glucocorticoid receptor, which is a protein which actually has binding site for this cortisol. So when this cortisol enters the cytoplasm, this glucocorticoid receptor binds to this cortisol. So binding this the glucocorticoid with the receptor actually makes it a dimer, or there is a conformational changes happens, and this actually turns into a dimer. So this is the dimer. Here we can see the dimer. So only after being converted to this dimer, it can actually traverse through from the cytoplasm into the nucleus. So this is the nuclear membrane. So this is the nucleus. So this dimer along with this hormone enters the nucleoplasm where the DNA is placed. So there are other elements which is called glucocorticoid response element, that is been depicted as denoted by GRE. So GRE is nothing but glucocorticoid response element, or that is the gene, particular portion of the gene that regulates the synthesis of this PepsiK. This dimer binds to this GRE and enhances the transcription of the GRE or that particular gene, so that mRNA corresponding to the production of PepsiK will be produced, and this mRNA then travels out to the cytoplasm where it is being translated to particular protein. So this is how what is called this is how hormone affects the PepsiK synthesis. Okay. And now we will see other regulatory mechanisms in which there are regions which we have already mentioned, that is called the enhancers, which are other than the promoter sequences, which actually have much role in gene regulation. So these enhancers may be situated thousands or thousands of nucleotide away from the promoter sequence. Okay, so this is the promoter sequence. This is the promoter sequence, and this the enhancer sequence. And this is the promoter, this is the enhancer. So this is being situated much away from this promoter. Still it has a role in controlling the gene regulation or the transcription. It is because even though it is being situated much away from this promoter sequences, it can actually controlled by it can actually control the gene transcription by some agents which are known as activators. So through these activators, these promoter, so because we know that this DNA is actually nucleosome model, so it is actually wound around these histone proteins. So that particular structure actually enhances this the transcription. So that thousands of thousand nucleotides away from this actually the promoters, the enhancers which actually situated 1000 nucleotide away from the promoter can actually come closer to this promoter sequence and regulate the synthesis of or the transcription. So this is how it is being done, and these enhancers are actually capable of rotating or at 180 degree, even though it is being rotated or at 180 degree, this the transcription, the efficiency of the transcription doesn't actually change. So this is what we confer from this. So this is we can see that here this is the enhancer and this is the data box of the promoter sequence, and these are the general transcription factors. So these enhancers are connected to the promoter sequence through connected to the general transcription factors through the promoter by means of some other proteins known as supplementary proteins known as co-activators, or it can directly bind to this, or it can directly bind to the promoter sequence and the general transcription factor. So the two-way attachment is possible. Either it can the enhancer can bind to this general transcription factors by means of co-activators, or it can bind directly to the general transcription factors. Okay. So that is about so we have seen all these about this the activation of a gene, and now we will see the negative regulation. We have seen the positive regulation, now we will see the negative regulation of gene expression. So how it is been the genes or the transcription is being negatively regulated or suppressed. Okay, so that is the main negative regulation or the regulation in transcription happens through methylation. So methylation or DNA methylation is a common feature in eukaryotic uh, nucleotides, and 1 out of 100 nucleotides be a methyl group, and this methyl group or methylation is actually a marker or a tag to show that it is different from that portion, or the activator portion is different from the other regions of a gene. So it is being situated or located at cytosine rich areas or GC rich areas. So GC rich island is the main region where this methylation is being usually occurring. So these are also being concentrated in GC rich islands, which are located near the transcriptional regulatory regions. Okay. So the one of the example of DNA methylation is of gamma globin gene, which is seen in fetal liver. Okay, so the methylation within the regulatory region of gene under activation tend to decrease. So usually the gene which is to be activated, that region will be devoid or will be lacking the methylation compared to the other region which should be deactivated or repressed. Okay, so the level of methylation in gamma globin gene in fetal liver is highly reduced compared to other tissues. So in other tissues, the methylation of gamma globin is higher when compared to the fetal liver, because the fetal liver, the production of gamma globin is essential for its development, so that the methylation is being highly reduced in that region. Okay, so how this methylation actually controls or inhibit a gene transcription? One is the methylation inhibit the binding of the transcription factors to that region, regulatory portions or the promoter, so that no transcription happens. And the second way is initiating the attachment of repressor proteins to the regulatory region, so that this transcription is ceased or stopped. So this is how this regulation, negative regulation, is being attained by DNA methylation. And another important feature is the structure of chromatin. As we have already mentioned, this DNA is actually a nucleus, and it is highly coiled structure, not in a linear model, and it is highly coiled, associated with proteins known as histone proteins. So these are the histone proteins, and it actually happens to see in a nucleosome model. So this is a nucleosome. It wound around twice on these nucleoproteins. Okay. So this is how an enhancer, this particular structure actually enhances the transcription by making this enhancer and the Tata box or the promoter region come closer, so that these will be attached by the co-activator, and hence it can initiate the transcription. Okay, so through this chromatin structure, even though this chromatin model, even though the enhancer is 1000 or 10,000 nucleotide away from the promoter, it can come very closer because of this model, and then it actually enhances or increase the transcription process. And there are some other proteins known as SW1 and SNF. These are multi-subunit protein complexes which can actually pry open this histone DNA model or DNA complex structure, and free up this region for transcription, free up the region of gene for transcription. And after transcription, it comes to the earlier conformation or this type of nucleosome model itself. Okay, so this is how this nucleosome model enhances um, the process of um, transcription.